Σιγά σιγά κάνω απλά την εισαγωγή στα ελληνικά, μετά μπορείς να μιλήσεις στα αγγλικά, Georgia. Καλώς ήρθατε στο σημερινό σεμινάριο. Welcome to our seminar. Έχουμε τη χαρά σήμερα να είναι μαζί μας η G.Papadogeorgou. Η Georgia λοιπόν έχει σπουδάσει στο μαθηματικό εδώ της Αθήνας και μετά έκανε μάστερ στη βιοστατιστική και κατόπιν διδακτορικό στο Harvard. Δουλεύει πλέον σαν assistant professor στο τμήμα στατιστικής στο Πανεπιστήμιο της Φλόριντα. Και σήμερα έχουμε τη χαρά να την έχουμε μαζί μας να μας μιλήσει για Covariate-informed latent interaction models addressing geographic taxonomic bias in predicting bird-plant interaction. So, Georgia, you may start your talk. Thank you very much for kindly accepting this invitation.
Πρώτα θα πω το ευχαριστώ μου στα ελληνικά. Ευχαριστώ για την πρόσκληση και ευχαριστώ που είναι μία από τις σπάνιες φορές που το επώνυμό μου το προφέραμε σωστά, γιατί όταν παρουσιάζω στα αγγλικά κανένα δεν μπορεί να το πει.
Έχω και επίσης εγώ πρόβλημα.
Ναι, το φαντάζομαι. Είναι και η πρώτη φορά που αναφέρει κάποιος ότι τελείωσε στο μαθηματικό Αθήνας, το οποίο και αυτό ήταν ευχάριστο, να θυμηθώ τα παλιά. Ναι, τώρα με συγχωρείτε που θα μιλάω στα αγγλικά, φαντάζομαι ότι όλοι μπορούν να καταλάβουν.
I guess it's fine to switch to English because you have some people not Greek as well. So.
Okay, I didn't realize it. Okay, yes, it's a lot easier for me to present in English because I only talk about my research in English, pretty much. So today, so feel free to interrupt me because I cannot see you. Jump in, just unmute yourself, or Panayoti, you can let me know if there's any questions. This is a project that I started during my postdoc with David Dunson at Duke and two wonderful ecologist collaborators, Carabello and Otto. This project, I hope that it will be finished this year, but it's a little bit different from what I normally work on. I normally work on causal inference, but I have also worked on Bayesian methods before, and I thought that this talk might be more fitting for the department here. So in this presentation, I will talk to you about modeling networks, and specifically about modeling ecological networks of bird and plant interactions. And I will come back to the motivation in a little bit. But from a statistical perspective, a lot of the measured networks that we have in the world are incomplete, meaning that we observe some of the interactions that might occur among nodes in the network, and we record those. But those measured interactions are not all of the possible interactions, and they might also be measured with error. So the measured networks are often incomplete, and sometimes some of the recorded interactions might not be truly present. Now, these incomplete measured networks might occur, for example, if we target specific individuals. So, for example, if we talk about networks, social media networks, we might follow who likes a certain individual's posts. And then we have focused on a specific individual and just measure what are the connections that this specific targeted individual makes with others. But a lot of times also there might be a large underlying network, but we might only have access to a subset of the nodes. So we might not be able to see what happens for nodes that are not in our target set. So even though we have incomplete measured networks in many different applications, our interest is to understand what is the network structure in terms of connectivity across the whole, perhaps large network. So we want to understand what is the true underlying interaction network from which we have sampled some of the connections with the potential for error. But also we want to understand in a lot of situations, what are the covariates that help inform whether an interaction is possible? So for example, two humans that have similar interests might be more likely to form an interaction on social media than not. But also people that have similar political beliefs might be more likely to like the same post or to like each other's posts. So we want to also understand which are the covariates that actually drive these node interactions, those interactions among nodes. these measured incomplete networks that we have available. Now, incomplete networks in general do not necessarily have to be considered a problem. So if we have measured data on a specific individual and our interest is to understand the connections of that specific individual, then the measured data that we have might suffice and might be representative of what we want to learn. But if the population we want to learn about is not the same or is not properly represented in our measured networks, then trying to infer what happens in our targeted population from these measured incomplete or measured with error networks might not work very well. So this might occur, for example, that if we follow, social media posts by well-known politicians, but we want to understand like what are the social network interactions of anybody, then since we have only followed well-known individuals, then the knowledge that we will acquire from those measured networks will not necessarily generalize to the target population that it has different characteristics than the measured population. So this is something that happens a lot in ecology. So here, like my motivation has to do with species interaction networks. So what that means is that there have been ecologists and researchers that have gone out in the field. I will be talking about Brazil. And there are researchers that have gone to different parts of Brazil and have observed species of birds and plants and have tried to record when a bird will eat the fruit or the seed of a specific plant. Now you can imagine that even if we put a lot of effort on sending people to Brazil to actually do this tedious work of keeping track of everything we observe in terms of species interactions, now these measured networks will still remain incomplete. One of the reasons why these networks, the measured networks that exist in the literature, are incomplete is that a lot of times researchers have a specific focus, and for example, they might focus on specific species. For example, you can imagine that understanding something that has to do with, lions or panthers might be a lot more interesting than understanding what a snail eats. So we have a lot more studies that might want to focus on the popular or interesting species rather than on the more, less exciting species. So a lot of the measured networks have this kind of preference of measuring interactions of specific species. And what those measured networks will look like if we were to kind of imagine that I have listed all possible birds on the left-hand side on the rows and all possible plants on the columns. Species-focused studies will follow only a specific subset of birds and try to understand what plants do those birds eat. It's like a recorded interaction is shown here in blue. Another study might focus on a different set of birds and might try to understand what plants these three birds over here consume in terms of consume. But there's also plant-focused studies that try to understand which birds or which frugivore populations eat the seed or the fruit of these specific plants. So you can see already that the measured networks that if we want to generalize knowledge from these networks to the general network of all bird and plant interactions, then that might not be easy to do since we have this preferential sampling of birds and plants in these species-focused studies. Now, yes. So I have taken, in my data, I have, I think 81 maybe individual measured networks. And I have information on whether those studies are animal-oriented or plant-oriented. So an animal-oriented is the one that focused on specific birds. The plant-oriented is the one that focused on specific plot for the unique number of bird species and the unique number of plant species in these studies. And we see that.
Can I ask a question here? Are these studies take part at the same region? For example, I don't know, in Brazil or?
That's a great point. That will be the geographical bias that I will show you in a little bit.
Okay. So I see.
Yeah, so they don't necessarily take part in the same region, and therefore they don't have the same species that are even available to be observed.
I see. Good. Please go.
Okay, so for these like animal-oriented studies, because they follow a specific set of birds, they have a low number of birds that they have recorded interactions for, but a high number of plants that they have recorded interactions for. And the reverse is true for plant-oriented studies. So plant-oriented studies focus on a small number of plants and try to record all of the possible birds that can eat the fruit of the specific plant. So we see that there's this imbalance in how many unique species animal and plant-oriented studies have information on. Now, and I will get to the network study in just a second. So network studies are it tries to solve this problem. Essentially, in network studies, researchers do not follow specific species, but instead they go out in the field and they record anything that they see. So these are the most representative in terms of number of species that are observed. So we see that a network study over here has a large number of unique bird and plant species. And generally what measured networks from these studies might look like, they might look like these types of networks where the white boxes that the interaction has not been recording and blue boxes the interaction has been recorded. What we see is that there's kind of this, the recorded interactions seem to be everywhere on these hypothesized tables up here. So the network studies seem to be great, they seem to be representative, but Panagiotis's point is coming up now that in fact, even though network studies seem more representative of all of the species that we're interested in, they actually are not, because all of these studies focus on a specific geographic area. And when the study focuses on a specific geographic area, so for example, a study that focuses near Sao Paulo might not, this area might not have a certain type of plant. And if since most of the studies are focusing here on this, on the near the ocean, plants and birds that live further away from the ocean and are not really represented in those regions will be underrepresented in those measured networks as well. So there's these two sources of bias. There's these two sources of bias in these measured ecological networks. One is more obvious than the other. So there's the focus of the study that might be bird, so animal or plant-oriented, but there's also this underlying geographical bias that makes some of the species harder to observe than others. So if I were to take the interactions that are measured across any of the networks and put them together in, one network, so this might be a picture showing all of the interactions that have been recorded across any of the studies, across all of the studies. So one might originally think, okay, that this is a relatively representative profile for bird and plant interactions, but however, that's not true because those secret biases, those biases of the individual studies do propagate here, and they lead to network of all possible recorded interactions to be biased as well. So even this combined network is taxonomically and geographically biased. So even when we take this combined network, we might have interactions that are not recorded, but those interactions could in fact be possible. So just because an interaction is not recorded, it doesn't mean that it's truly impossible. And this is exactly what we want to learn. So we want to understand, how do species interact, which species can interact with each other, while adjusting for these biases in the individual studies. And we also want to learn which covariates are most important in driving these species interactions. So the motivation of this work, and I showed you a plot of Brazil already, is that there's this very big forest. It's called the Atlantic forest. It spreads mostly in Brazil, but also parts of it are in other countries, in the nearby countries. But the Atlantic forest currently includes only 12% of its original biome. So its biodiversity is decreasing really quickly. So what happens is that plants that live in that area rely on species populations that consume the seed or the fruit of the plant for seed dispersal. So the idea is that if a plant wants to survive, we would like the plant to be consumed by birds, for example, and then the bird will travel with the seed and drop the seed in a different area so the plant can propagate and start growing in that area as well. But the reductions that we see in these, for example, bird populations might lead to disruptions in which seeds, which plants are dispersed and which are not. And this also then leads to disruption in this regeneration of the Atlantic forest. And we expect that these issues that have been observed will only get worse as climate change and deforestation is occurring in that area. So our motivation from an applied perspective is to understand how these biological communities of birds and plants will respond to these environmental changes, and understand what would happen between birds and plants for any pair of bird and plant, because there might be a pair of bird and plant that do not coexist right now, but as the forest changes, those birds and plants might find themselves exist in the same area, and they might suddenly find themselves in a situation possible for an interaction. So we want to understand, would a bird consume the seed of a given plant if they coexisted and they were given the opportunity? So getting to the statistics slowly. So, okay, I remember the number wrong. We have data on 85 individual studies. 19 of them are animal oriented, so they focus on specific bird species. 45 are plant oriented, and 19 of these are network studies. The remaining 2 studies are a combination of network and species oriented study. So we have 232 birds, 511 plants. So I use I for bird and J for a plant. And we also have these measured networks across these 85 studies, which we use capital A to denote the recorded interaction. So if AIJS is equal to 1, it means that bird I was recorded to interact with plant J in study S. We also have measured characteristics for birds and plants. So XI includes the size of the bird, the beak size of the bird, so how big its mouth is in a sense. WJ includes plant information such that the size of the seed and the color of the plant and information like that. So again, in terms of how to rephrase this in statistical terms, what I would like to learn is the bipartite network that underlies these species interaction. So I want to understand if this underlying L matrix that is no longer study specific and it just tells me whether bird I would interact with plant J if given the opportunity. So if that interaction is possible, Lij will be equal to 1 and 0 otherwise. And we want to take into account the fact that unrecorded interactions are not necessarily impossible. So unrecorded interactions might be possible. And we want to take into consideration that these recorded networks are prone to biases because of study focus and study geography. At the same time, we want to understand covariate importance in these network models that we will be using that will be based on latent factors. So we want to figure out how can we study covariate importance in these latent network models. So our measured data are in the form of this A array. So essentially this A array is of dimensions 232 birds, 511 plants, and 85 studies. We have this X information about bird characteristics and W for plant characteristics, and we want to somehow elucidate a likelihood for our measured data, our data. So to do this, we will introduce latent factors. So we think that for every bird I, there's a set of characteristics U1, U2 up to UH that now we can think about this for now as just a set of characteristics that are encompassing the bird's behavior. Now, these can be arbitrarily close for now to the covariates X, to the measured covariates, but we can think of this as like a lower dimensional representation of X that might also try to capture parts of the bird's behavior that is not part of the measured data. And similarly for plant J, the latent factors Bj are supposed to represent the characteristics of a plant that will play a role for the plant's interactions and also the plant's detectability. And they can also be thought to be arbitrarily close to this, to the covariates W. Now, I don't know if this will be, I assume that everybody has, or not everybody, but maybe most people have seen latent factor network models. But if you haven't, I like to think of this U and V as if I had X and I was doing PCA on X and I was taking the first few principal components and adding a little bit more that might not be captured in X directly. So I think of X and W as something I'm thinking of U and V as something that is relatively low dimensional, that is trying to capture all of the important information for these birds and plants. Now, when we come up with a likelihood, I'm sitting at a place that there's exactly sun in my eyes, so I'm just slowly turning. So when we want to write down a likelihood for all of these data, if we focus first on the data of the measured networks, then we have to take into consideration the following: that if we want an interaction Aij S, if we want an Ij interaction to be recorded in study S, all of the following have to happen. The species have to occur in the study area. The researchers need to be interested in those specific species. Those species need to be possible to interact, and that interaction has to be detected by the researchers and then recorded. So all of these have to occur in order for an interaction between plant I and J, bird I and plant J, to be recorded in study S. So what plays a crucial role here then is to understand what are the species that occur in the study area, which we can consider them fixed here using published maps of species co-occurrence, or we actually use a data-driven way to decide this OIJ. I can talk to you about this later if you want. We also need to understand what is the study focus, which we actually have direct data on. So we need to understand whether study S would record the IJ interaction if that interaction was observed. So if study focus will be equal to 1 for all species if we have a network study. But if we have a species-oriented study, so if, for example, we have a plant-oriented study, this F matrix, or this F matrix for that specific S, would only have ones at the columns that correspond to the plants that are in focus. So this study focused array is important for addressing the taxonomical bias of the individual studies. But then there's like this other information that plays a role on whether an interaction is actually recorded. So it matters whether the interaction is possible, which is something we don't know. And here this little F shouldn't exist. This LIJ is exactly what we're trying to learn. We're trying to, but we need to include this LIJ when we come up with a likelihood for A. But also, different birds and plants might be harder or easier to observe. So you can imagine that if you're in a dense forest, a plant that has a bright red flower or a bright red fruit will be easier to detect than a plant that might have a smaller and less bright of a color of fruit. So species detectability is something that will also play a role on how likely it is that we record interactions of specific species. And we need to take all of this into consideration when we elucidate a likelihood for the measured interactions. So in fact, I know that this is somewhat a lot of notation, but what we want to think about is when I'm trying to come up with a likelihood for the measured networks, I need to be essentially conditioning on whether that interaction is truly possible, whether the study focused on those species, whether those species co-occurred, their detectability, but also the species information, either the latent information or the measured information. So again, when we do this, there are going to be dependencies across the measured networks. I'm not going to go through all of them again, but essentially, like if a species, let's focus on the last one, if a bird is hard to detect, then it will have a low prevalence across all of the studies. Because if a bird is hard to detect, then it will be hard to detect for researchers in all of the different areas and all of the different study focuses. So that will create species detectability, creates dependencies across the measured networks. But once we condition on species co-occurrence, study focus, and the possibility of true interactions and detectability, then we can make a more reasonable conditional independence assumption on those measured networks, which allow us to write now these measured interactions as a product over bird, plants, and studies of individual conditional probabilities. So again, this kind of like, we make a conditional independence assumption that is more detailed in the paper that allows us to go from the joint distribution of the measured networks to this individual, so pair study specific conditional distributions. And the way we specify this conditional distribution is to say, okay, if an interaction, so let's think of L, F, or O, L, F, and O all being equal to 1, what this tells me is that the interaction is possible, the study focused on that pair of species, and those species co-occurred. So if all of these occur, then how likely is it that I record an interaction? It's as likely as the product of the species detectability scores. So this equation here is the one that tries to account for this taxonomic and geographical bias by explicitly saying that if a study did not focus on the species or the species did not co-occur, then it will be impossible for that interaction to be recorded. So we are encoding this preferential sampling of the individual networks in this likelihood over here. And I don't want to focus too much on this, but all I wanted to show you is that once we do this, the likelihood essentially for the measured the conditional distribution of them for the measured networks factorizes in these two components. And essentially the only component that encodes any amount of information for the true for informing the parameters of our model afterwards comes from those pairs that exactly are possible to be recorded. So if a pair of species is not the focus of the study, or it's not, or a pair of species do not co-occur at a specific study site, then it will not really be informative of any of our follow-up parameters. So we are essentially saying that we need to only use information that comes from pairs of species that are possible to be observed in an interaction, rather than using all of our measured networks that have these underlying biases. So these biases are, so we are kind of controlling for these biases by saying only use information that comes from the parts of the network that do not have that bias, because that interaction would have been observed if it was possible. So we need to specify now distributions for all the other quantities as well. And actually, I'm going to go through this quickly, and I'm going to show this to you schematically afterwards. Essentially, we assume that this true underlying interaction network is a latent factor model, saying that these latent factors are the ones that are of the birds and the plants that are the ones that are interacting, to tell us how likely it is for an interaction to be truly possible. And also we assume models of the species detectability scores that only use these latent factors. Now, I will say this here, and I will show it to you in a second. Essentially, even though this conditional distribution that we specify here is conditioning on measured and latent characteristics, so this measured X and measured W, but also the latent U and V, we actually make a conditional independence assumption here that says that the measured characteristics do not play a role for this network model once we condition on the latent characteristics. And the same kind of conditional independence assumption happens for these detectability scores, where we say that if we know this latent factor that, remember, is supposed to encode all of the useful information in the measured covariates, then what is left in the measured covariates does not really play a role for these detectability scores. And the reason why we call our approach covariate-informed latent factor model is this last part, that we want to essentially allow these latent factors that we are using in our detectability model and in our network model. We want those latent factors to be informed by our measured covariates. And we do this by also assuming models for our measured covariates that depend on these latent factors. So this relates, or in a sense, relates our latent factors back to, if you think about this, back to PCA, because we're thinking that these latent factors are the ones that are containing the crucial information in our measured bird and plant characteristics. So essentially, if I were to kind of put this in a figure, what we're saying, so here the gray nodes, and I have excluded the parameters in this graph. Is supposed to be just to build intuition rather than to show a complete model, but the gray nodes are not observed and the white nodes are observed data. So what essentially our model is assuming is that the measured characteristics X, I, and WJ are not directly informative of the measured networks A, or of the detectability scores Q and P, or of the truly possible interactions L, but the relationship between the covariates and all this detectability recorded and true interactions comes through the latent factors U and V. And essentially, we are not specifying a network model that depends on covariates directly, but we specify a network model that depends on latent factors, which are informed by these measured covariates. Now, I'm going to just keep the prior specifications or the the sampling scheme. We use pretty standard tools. We use MCMC for approximating the posterior distribution. Most updates were done using Gibbs or Metropolis Hastings, but because we also had logistic models, we used the polyogamma data augmentation for some of our parameters. And we performed a number of different simulations to investigate how our model performs in comparison to a network model that uses covariates directly, that puts the model to depend on these latent factors. How would a model perform if this network model depended on covariates directly? So covariates were included in this linear predictor of the true interaction model. And we consider a number of scenarios, mostly because reviewers wanted to see more scenarios. And so there were like 24 different data generative mechanisms that varied many different things, whether we had the same or different covariates that were informing the different submodels of true interactions, detectability, and detectability, whether we had the important covariates being in the set of measured covariates or unmeasured covariates, the correlation between the covariates, and also whether we were in high or low information scenario being represented by the proportion of pairs that have F times O equal to 1, the proportion, essentially we're talking about the proportion of species pairs that are possible to be recorded in the study. So, and we also tried a number of different approaches, but here I'm only going to show you one simulating scenario and only two approaches. So our approach that uses latent factors and the approach that uses covariates directly. And this is essentially what we saw across all of the simulation scenarios we considered. So here I'm showing you the area under the curve for our ability to predict the truly possible interactions. And throughout our simulations, we saw the latent factors in general have higher predictive ability for missing interactions, but also we see kind of this interesting trend that the more opportunity we have to observe an interaction, so the more studies with F times O equal to 1, the higher our predictive ability for understanding whether an interaction is possible or not. And that makes sense, because if an interaction is not measured, so it's unrecorded, if we have a pair IJ that have had the ability to be recorded in many studies, but that interaction was not recorded, it is very likely that it's because that interaction is truly not present. So by taking the taxonomic and geographical bias into consideration, we actually see that this propagates, and our model is able to learn that that an interaction that was possible to be recorded across many studies that was actually not recorded is most likely not possible. Okay, so we also created an approach for understanding. Now, remember that in our network model, we have latent factors informing the possible interactions, but we assume that covariates are not informing the true interactions directly, but they only inform whether an interaction is possible through these latent factors. So since we are not just including the covariates in the linear predictor of our interaction model, we cannot look at a coefficient to understand covariate importance. So we cannot look at a coefficient to understand how important is the plant color in forming interactions or something like that. Now, the fact that we're not including covariates in the interaction model is actually by design. And this is kind of like where my causal background comes into play, because including covariates in the interaction model and trying to use their coefficients to inform covariate, to study covariate importance is actually flawed philosophically. And I can talk to you forever about this if you want. So essentially, the fact that these covariates are network structures, so similar species will have similar value of those covariates, but also because the latent factors will be similar for genetically related species, creates some multicollinearity between our latent factors and our measured covariates that does not allow us to just simply interpret coefficients in our interaction models for variable importance. And there's recently some work from the ecological literature that is also studying this. So should I be stopping in, like, how much time do I have left?
Let's say 7-8 minutes.
Oh.
7 to 8 minutes, you can, it's fine.
Great, awesome. Then I will quickly maybe, so we have these, so I will probably spend a couple of minutes explaining how we study variable importance in this latent factor network model, where we use the covariates to inform the latent factors through a separate model. So you can think about this in terms of our MCMC. We have, let's say, capital R posterior samples of our MCMC, and at each iteration of our MCMC, we have the probability that for a true interaction between bird I and plant J. So I denote this as little Lijr. So this is the logit of the probability for an interaction between bird I and plant J at posterior sample R, once these taxonomic and geographical biases have been accommodated for. And then let's think about trying to understand, how does the kth bird covariate inform, how much does the kth bird covariate matter for forming interactions for the bird species. So we have, though, this probability of interaction for all birds, showing again in the rows, and all plants showing in the columns. So we create a test statistic that looks at the correlation between our probability of interactions and that bird covariate. And it does this for each plant species, and averages across these plant species. And then we also take the average across the posterior samples. So in a sense, this is a measure, this T observed is a measure of how correlated is our bird covariate, our measured bird covariate, with the probability of an interaction for birds across the different plant species. And then we can permute the covariate vector X and perform the same procedure. And by permuting this covariate vector, we break the relationship between X and the probability of interaction, and we can acquire, in a sense, the null distribution between the covariate and the probability of interaction. And we can compare how far our observed test statistic falls from this distribution of the test statistic under the null, that there's no relationship between the covariate and the probability of interaction. So for a Bayesian person, perhaps, like, I don't know, you might be getting goosebumps, because this isn't Bayesian, right? But we are using a permutation-based procedure to understand variable importance. But what we have seen in practice is that this approach performs really well. And I'm not going to get into the simulations, but if you're interested, I'm happy to talk to you about this afterwards, or it's also in the paper. So when we fit this approach in our study, we got probability of interaction for species that might look like this. So here I'm only showing a subset of the species. and I have organized the species by their taxonomic family. So species that are within these big rows are more genetically related. The species that are across different categories on the left, and also same for plants. Here we have 1, 2, 3, 4, 5, 6 taxonomic families for plants, and then individual plants that are within each one of those families will be more genetically related than plants that are across these different families. And what we see is that based on our model, we have relatively taxonomically we seem to get predictions that make sense. In the sense, for example, let's look at this part over here. We see that species that are within the same family tend to form more similar interactions compared to species in different taxonomic families. So it seems that our model is able to capture this underlying structure, that species that are the genetically related will also act in a similar fashion and consume plants of similar types.
Georgia, sorry, the dots here represent observed interactions. Am I wrong? Or?
Correct. Yes. So the black points are the recorded interactions. So this was a quite sparse network. We only have about 3% of the possible pairs that have a recorded interaction.
I see.
So compared to like what a model based on these are the results based on a model that uses covariates directly, and we see that a model that uses covariates directly performs really poorly, in the sense that it shows these like all or nothing kind of interaction profile. So for example, it shows that plants in this family essentially interact with all the birds, whereas some plants in here, perhaps they interact with none of the birds. And that's because the model does not allow for flexible relationships between the covariates and the interaction profile, but instead specifies how that relationship occurs specifically through a parametric model. Now, perhaps different specifications of the parametric model would perform differently, but we would have to be careful in this model about how we actually specify it. Whereas our latent factor model, we don't really need to specify how those latent factors play a role in the network construction. Now, the last thing I want to show you is this the covariant importance metric based on our approach. So we can order our covariates from most important for forming interactions to least important for forming interactions. So at the top here, we show the bird covariates, and at the bottom is the plant covariates. And the ones that have star, I'm not sure that's really easy to see, are binary covariates, whereas the ones without the star are continuous covariates. So we focused on the most important continuous covariates, which for birds was the body mass of the bird, so essentially the size of the bird, was identified based on our algorithm as the most important continuous covariate. And for plant species, the most important continuous covariate was the fruit diameter. So the size of the fruit for the plant and the size of the bird seem to be the most important covariates for which interactions are possible. And again, that makes sense. And then we took those two covariates and we reordered our species in terms of increasing fruit diameter on the x-axis and increasing body mass on the y-axis. And then we plotted the probability of interaction based on our model. And we found that so green means higher probability of interaction, whereas orange means lower probability of interaction. And we see exactly what one would expect, is that if we have a small fruit diameter by birds of essentially any size. And similarly, large birds are able to consume plants and fruits of any diameter. However, we see at the bottom right that essentially very large plants are most likely to be consumed by large birds. And that is expected because a small bird will not be able to consume a big fruit. So our model is able to capture this interactive relationship between body mass and fruit diameter without explicitly having to specify that these two covariates are expected to interact. So this is where I'm going to wrap up and give some final thoughts. So we introduce a network model based on latent factors that uses measured networks, incomplete measured networks, so the measured networks that might have some biases. And part of the contribution of our work to the literature, to the statistical literature, is to propose including the covariates in these models through separate modeling of the covariate latent factor relationship, rather than including the covariates directly in the network model. And we proposed the variable, kind of like a permutation-based approach for understanding variable importance in these covariate-informed latent factor models, latent network models. Now, in terms of our application, we were able to complete the bipartite graph of species interactions in a way that incorporates this taxonomic and geographical bias that the individual studies have. And we, based on our ecologist collaborators, we received estimated interaction matrices that were more reasonable compared to competing approaches. Now, we have had some discussion about extending our work to simultaneous model co-occurrence, species co-occurrence, and interactions. That would be great, especially because species co-occurrence is a hard problem on its own. So you can imagine that whether species co-occur will be related to whether species are possible to interact. Because, for example, a bird will not choose to live in a certain geographic area if there's nothing in that area that the bird can consume. So simultaneous modeling of co-occurrence and interactions can actually help us learn both of these processes better. All right, so I'll just stop here. And again, thank you for the invitation.
Thank you, Georgia, for the interesting talk. Are there any questions for Georgia? Can I ask a question, Georgia? Do you have any, I don't know if you mentioned that, but at some point I saw something that the factors of the model, you have problems with the identifiability, right?
So of course, like the latent factors are not identifiable.
Okay, so this is due to the intrinsic nature of factor models because you have rotational ambiguities and so on, or something else?
No, that's exactly it. Because and also like the scaling of the latent factors, you can scale U and scale, yeah. That's why. But and we could restrict the latent factors in some sense. Like a lot of people, what they do is they set the latent factor of the first species to 1 and then condition or something like this, and then conditional. And if they fix the latent factors for a single unit, then the rest of them are now identifiable. But because we don't try to interpret our latent factors, we don't really care about this. And trying to make the latent factors identifiable in like a Bayesian model might also make you, like, not might, but it will make probably the MCMC a lot harder to implement and perhaps to also converge. And sometimes having these unrestricted latent factors might make your MCMC converge faster.
Yeah, so that's really interesting because I've done some work in the past working with the interpretation of latent factor models. We can do this after MCMC, so you can have a MCMC running, and then you can post-process with various things. So that would be interesting. It's good to know that these problems occur in your case as well. So what about the number of factors? How do you choose the number of the dimensionality? I mean,
I just chose so I looked in simulations about the impact of the number of latent factors, and I saw in my simulations that generally like I only had like 3, 4, 5 ever used because I have a shrinkage prior on the coefficients of the latent factors. So I specify my H to be 10, and after the fact I make sure that enough of them have been zeroed out often in my MCMC that allows me to say, okay, even though I specify 10, the effective number of latent factors used was 3 or 4.
I see. Okay, no, that's fine.
I don't know though. I'm not completely sure about the identifiability. I'm not perfectly certain. So I'm aware of identifiability issues mostly in when we use latent factors for modeling multivariate data, but I'm not sure how you solve identifiability in latent factor network models. Now that I'm thinking about this more closely, it's like it might be more complicated here because I'm not sure whether rotations of U would lead to the
same likelihood, let's say.
Right. We might have problems of identifiability in terms of simultaneous rotations of U and V.
Yeah, I guess that's the case. Yeah, because you have both of them. But it would be interesting to think about it. Yeah. So, are there any other questions for Georgia?
Maybe just a quick one. I like the definition of the aim for getting a measure of variable importance. I wonder how you would go about try to define something like that in a nonlinear model, perhaps, because averaging may be tricky, I suppose. Or I don't know. Would you have a sense? Or, where you average and then you permute and you get a measure of it. Yeah, exactly.
Yes, so I also tried using the maximum of these correlations as a test statistic in my simulations. I think this is what you're asking about, like about averaging across the species in this.
Yeah, would you sort of amend this if you had a nonlinear sort of term somehow? Just wonder because averaging over a nonlinear case may be tricky.
So I haven't thought about this before, but my thought process right now is that my latent factors U and V are assumed to be normally distributed. Now, if we were, and that kind of creates some nice structure around these logistic probabilities. I wonder if I think this might have to do more about non-symmetric distributions of our predictors in our logistic model. So I'm thinking that perhaps because my U and V are normally distributed, doing something like an averaging of these probabilities makes sense, that it works well because everything is symmetric, everything is nice, so averaging kind of brings us to the middle. But if what I put in my model, if the predictors that I had in my model had these long tails in one direction, then using a test statistic that tries to look at those explicitly, like what happens at those tails of the covariates, might perform better. So I'm not sure it has to do, and having interactions or having higher order terms in your network model would essentially bring you to this scenario. Like if I had a normal covariate that was a predictor in my network model, but I thought that that covariate squared was part of my logistic model, then the fact that that covariate was squared in that logistic model would essentially bring us to a chi-square distribution that has a long right tail. So we might want to use a different test statistic. So I think you're right, but I had never thought of this before. I don't know if that really, I don't know. I wonder what you think about this, but that's where my brain takes me.
Yeah, no, I mean, it's not obvious, I don't think, and I wasn't sure how to, because I think it would be model dependent somehow, so I don't think it's trivial to do. You know, I think of it depends on the model, I'm guessing, but.
Right, I guess so too, but because here I use the logit of these probabilities, and the logit of these probabilities are essentially U times V. And because U and V are independent normal, then I think that these L quantities that I use for this variable importance metric are nicely behaved in terms of having something that resembles a normal distribution. And I think that that's what makes the averaging work best. But if what I put in my interaction model, or if I didn't use the logit of these probabilities and I use the probabilities directly that involve the X bit of the linear predictor, then the averaging might not be what's ideal to do. Which I think comes back to what you're saying, that greatly depends on what does the distribution. I think it depends on what does the distribution of your L look like conditional on those predictors. And if conditional on those predictors, that distribution of L is normal, then averaging works great. If conditional on those predictors, the distribution of L is something that has a long tail, for example, we might want to use something like a maximum for creating a permutation procedure.
Good. Thanks a lot.
So I guess that's it. Are there any other questions? Let's thank Georgia once again for the interesting talk. Georgia, all the best. I hope that we can, who knows, next week maybe in London.
Sounds good.
Okay.
Thank you so much for the invitation. It was great to speak in Athens. This is the first time I gave a presentation in the math department once. It was very interesting to give a presentation in front of my old professors. So thank you. I really appreciate it.